Welcome to Infectious Disease Lesson 4.3, How do Bacteria Adapt to Become Pathogens? The Adaptation Auction. The goals of this lesson are to reinforce how bacteria use structures and adaptations to survive in select parts of the body, design an ideal pathogen for an assigned body niche, and illustrate that maintaining adaptations has a cost, energy. By the end of the lesson, the students should be able to name adaptations and explain how they benefit bacteria in the right environments, describe how adaptations are used to gain access to nutrients, and discuss the limits to acquiring adaptations. We will achieve these goals with an adaptation auction, which involves working in teams to design a pathogen that will successfully live in a body niche. Students will compete with other teams as they bid on the adaptations. To prepare for this lesson, you'll need to review the key scientific concepts. Pathogenic bacteria have tools, adaptations, that allow them to overcome specific host barriers. The cost of maintaining adaptations limits the number of tools a given bacterium can have. You can review the scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure, and the student workbook provides additional explanation for students. Note that an annotated version is also available for teachers. Be sure to print the lesson worksheet. The key points of the do now are that pathogenic bacteria have adaptations to overcome host immune barriers, and pathogens have to be camouflaged to avoid the immune system. We'll get there by reviewing the adaptations and structures seen up to now. The do now begins by having the students consider what microbes need to become pathogenic. As we have seen throughout the course, microbes that are pathogenic have tools to overcome immune barriers. Before proceeding to the adaptation auction, take a few minutes to review the bacterial structures introduced in Unit 1, Lesson 3. The key points of the activity are that pathogenic bacteria have adaptations that allow them to overcome specific host barriers. The cost of maintaining adaptations limits the number of tools a given bacterium can have. We'll get there with the bacteria adaptation auction. Students will be divided into teams, and each team is given a habitat or niche in the body. For example, one team could be assigned to the lungs, while another team is assigned to the intestines. The goal will be to design a bacterium with the best chances of survival and reproduction for their niche. Then there will be an auction, and the teams will bid on the best adaptations for their bacterium. First, break students into groups of 2 to 4, and assign each group a body habitat. Note that if you have less than 4 groups, it's important to assign niches that will need to compete for adaptations in the auction. If the groups are not trying to win some of the same adaptations, the auction will be ineffective. To ensure that you assign competing niches, use the table in the student worksheet to select the niche. For example, microbes in the skin, nose, and lung will need similar adaptations. Next, describe the goals of the auction. Give the students 10 minutes to review the list of adaptations on the worksheet to choose the ones they want for their niche. When doing this, they should consider which adaptations will be the most important for the survival in the environment, and which adaptations to keep away from the other teams, and why. Each group only has $1,000 to bid with, so they need to strategize about how much money they are willing to spend on their favorite adaptations. They should also consider how much they would spend to keep other groups from getting select adaptations. Note that looking at the characteristics of other bacteria that share their habitat or niche on the table in the worksheet might be helpful. Now that the teams have designed their perfect pathogen, it's time to bid on adaptations. When going through the list of adaptations, it might be helpful to assign a student to record the accepted bids on the board. This way, each group can see how much money is left and which adaptations each group has. Depending on how much time you have for the auction, adjust the increments of the bids. For example, you may increase bids by $25, $50, $100, or $200. Because this is a unique lesson, it might be difficult to imagine how this lesson will run, so we have embedded a video clip from a classroom after we introduce the adaptations. In the next few minutes, we will briefly talk through the adaptations. For more detail about the adaptations, please refer to the teacher manual. Low replication rates help infections persist for long periods of time because they don't activate the immune system, whereas high replication rates allow bacteria to spread quickly within the host. Gram-positive bacteria have a thick cell wall that contains multiple layers of a sugar peptide polymer called murine that is a rigid molecule. Many gram-positive bacteria can withstand high salt on the skin. Gram-negative bacteria have a cell wall with thin murine layer but contain an additional outer membrane. The outer membrane has LPS, an endotoxin that causes inflammation. Gram-negative bacteria are also more resistant to bile salts in the GI tract. Acid-fast bacteria have a cell wall that also contains large amounts of waxes as well as murine. The cell wall allows the bacteria to resist the chemical barriers of the immune system. Capsules provide perfect camouflage from the immune system. Flagella allow bacteria to travel to find nutrition. And spore formation allows some gram-positive bacteria to survive when nutrients are depleted. Extracellular replication allows bacteria to spread rapidly. They may survive and spread on the body's surfaces and even on inanimate objects. Intracellular replication allows bacteria access to the nutrients they need from inside host cells and also to hide from immune cells. Bacteria can produce exoenzymes, a type of exotoxin. This can kill host cells, allowing them to spread into deeper tissues. Bacteria can also produce an endotoxin that causes inflammation, helping them spread into deeper tissues via the bloodstream. Some exoenzymes break down the connections or extracellular matrix surrounding host cells. For example, some exoenzymes can digest blood clots and help the bacteria invade wounds. This allows them to spread into deeper tissues. The adaptation auction may go a lot more smoothly if you watch the video of a classroom doing the adaptation auction. You can watch this video on the Great Diseases website or on our YouTube page. The key point of the wrap-up is that there is a cost to maintaining adaptations that limits the number of tools a given bacterium can have. In the wrap-up, students will consider what limits the number of adaptations a bacteria can have. The main idea here is that just like in the auction, there is a cost to bacteria for maintaining adaptations, and this cost is nutrients and energy. The key point of the homework is to review how each specific adaptation benefits the group's bacterium. We'll get there by having students complete their worksheets either in class or as homework by answering the following questions. First, to name their bacterium, describe their bacterium's niche, draw their bacterium, explain why their adaptations will help their bacterium. Note that they will do this based on the adaptations they wanted, not the ones they won in the auction. One of the most common questions students have is what happens if a group does not get the adaptations that they want. The point of the lesson is to pick adaptations that make sense for the assigned niche. The grading will be based on the adaptations each group wanted, not the ones they actually won in the auction. Another question students ask is, can viruses release toxins? The answer is no, only bacteria release toxins. This is the last lesson that focuses on bacterial pathogens. The next few lessons move on to viruses. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.